So today's question I have taken from Biochemistry Gate Life Sciences question paper 2019. This is question number 17 from that year. So this question states, okay, so yeah, welcome you all. So, this is question number 17, which I have taken from GATE to 2019 Life Sciences question paper from Biochemistry section. So, this question states the absence of detectable protein expression upon blunt-ended mutation-free cloning of an E. coli gene with its own promoter in E. coli cells can be due to. Okay, so there the protein is not being detected, and the cloning that has been done here is the blunt-ended mutation-free cloning. Okay, so it is written blunt-ended cloning. Blunt-ended cloning means like if you have a desired clone. So if you have a clone, okay. So this is question number 17 from GATE Life Sciences 2019 paper from Biochemistry section. So question states that the absence of detectable protein expression upon blunt-ended mutation-free cloning of an E. coli gene with its own promoter in E. coli cells can be due to. So the protein is undetectable. So upon blunt-ended mutation free cloning. So this cloning, they are saying it is a blunt-ended cloning. So blunt-ended cloning means, so in cloning you have a vector and an insert. Okay. So if you are doing a blunt-ended cloning means the cloning we do with the restriction digestion. That means that restriction endonuclease is used to digest the our particular desired vector or insert. So some okay, so some restriction endonuclease creates this kind of end that is called sticky ends. Okay, so this kind of ends, so vector also harboring same kind of end. So if the vector also have the same end, so this kind of ligation can take place. This end can be ligated, and similarly with this side, these sticky ends can be ligated. Okay, so this is sticky end cloning. Okay, it is producing some kind of sticky end where according to the complementarity of the sequences forms a new hydrogen bonds and get ligated and forms a complete construct. But in the blunt end cloning, the fragments generated after restriction digestion are blunt ended. Okay, blunt ended. Okay, so for example, if I'll digest, I'll give you an example of uh, sticky end and blunt end. So if I suppose I digest my sequence with BAMH1 restriction enzyme, so BAMH1 having the sequence of this CGA TCC. Okay, and this is the cutting site. Okay, so it will generate ends like this. One end it is generated like this. Like this. So this is a sticky end. Okay. Another end is other end which will be generated will be like this. Okay, so this is the sticky ends. When the complementary ends will meet, then only the cloning will take place. Okay. These are the complementary sequence which will go and attach here. Okay. So if sticky ends, the cloning probability will be more sure. Okay. Only the complementary, once the complementary ligation assembly will happen, then only it will get ligated. But in the blunt ended cloning, okay, so blunt ended, if I'll give an example, if I give you example, blunt end enzyme, if the sequence is like this given and this is not being cut like this but being cut in the middle somewhere, and it is generating a sequence like this. Okay, then no complementary, these are the blunt end that are being generated upon the restriction digestion. So upon the when you are putting the ligation, you can just ligate these ends by phosphodiester bond creation. Okay, and nothing complementary base pairing is required. So this kind of blunt ended cloning may be non-directional, or it is a non-directional cloning. So if suppose your starting is here and start is here and stop is here, so because of the blunt ends which are being generated, it can attach in either this orientation or in this orientation. So any orientation it can attach. Okay, so the protein is being not expressed. So first option is cloning occurred in reverse direction. That is a possibility that it can get the cloning has been done in the reverse direction. But even though it is in the reverse direction, the sequences will alter. The amino acid will be produced, will be different, but it is not like the protein will not be expressed. Protein will be expressed, the sequence, but the sequence will be changed. So this option is wrong, that the cloning occurred in reverse direction. Even in the reverse direction, the protein will be expressed. Second option is cloning occurred out of the frame. Out of the frame in the sense means this is my sequence. So, this is my sequence. Okay, so we have the codon like this. Okay, only in this sense, if correct frame will be maintained, then only it will read and correct amino acid will be synthesized and correct protein will be synthesized. If out of the frame, if the cloning has occurred out of the frame, then what happened? Then also, like if it starts from the second one, then this, so some other amino acid will be produced. Okay, or in this, okay, some other amino acid will be produced, but the protein will still be expressed. But still, even though out of the frame, the protein will be expressed, but here they, or it is a possibility that it may not express, but they have said that it is a mutation free cloning. That means out of the frame possibility is gone. Okay, so cloning occurred out of the frame, so that is also not a correct option because it is mentioned mutation free cloning. Third option is the codon bias. Codon biasness we have because of the codon degeneracy. Codon degeneracy means more than one codon is coding for one amino acid. So the uses of the codon differs from species to species. Suppose one codon which is in the gene sequence is being used, like if I'm putting a human protein in a bacteria, so inside the human, the in the gene, the codon is there. For that, the tRNA are not preferable. So bacteria doesn't use those codons and for in their machinery to synthesize that one particular amino acid. It uses some other codon. So that is why the synthesis or the production of the protein will not be there. But here it is clearly written that it is a E. coli gene with its own promoter in E. coli cells itself. Then the probability of codon biasness will go. So the third option of codon bias is also incorrect because it is happening in the same system. Fourth option is rapid degradation of expressed protein. Okay, so it is saying that cloning has been done. Okay, and it is maybe in the correct orientation. It is a mutation free cloning. It is not due to the codon biasness. The protein is being expressed, but as soon as the protein is being formed, the protein is being degraded. Okay, so that is a possibility that even the protein is being formed, it is being degraded, and because of that we are not able to detect the protein expression. So this is the correct option or correct possibilities for to explain this particular phenomena. So in absence of reticular protein expression upon blunt-ended mutation-free cloning, the fourth option is correct, that protein is being formed, but there is a possibility that it is being degraded, and because of that we are not able to detect the protein. So this is the correct option. And this is NPTEL reference where you can study more detailed about cloning and the techniques and their confirmation and the orientations about the cloning technologies. Thank you.